Good afternoon, everyone. I'm Asmita Khaniya, and I work under Professor Nadia Hirankan. I am a postdoctoral researcher from Faculty of Medicine, Chulalongkorn University, and I'm also a team member of our cellular immunotherapy research unit's CUCIT lab from Chulalongkorn University. So this talk will be a little bit different from the morning session, and we will learn little bit more about what is CAR T cell, and I'll be talking on the advantage of bidirectional promoter-driven dual chimeric antigen receptor CAR construct. So let me give a brief introduction. What is chimeric antigen receptor CAR T cell? The CAR T cell is a genetically engineered T cell, and it has to enhance the ability of T cell and recognize and kill the cancer. It has the single chain variable fragment, which is derived from the antibody, and it is specific to the tumor marker. It also has the, I'm sorry. So it also has the hinge domain, which confers the flexibility to the scFv. Likewise, it also has the transmembrane domain that anchors the ectodomain and the endodomain of the CAR construct. It also has the co-stimulatory domain, which is derived from the CD28 or 41BB. And it also has the intracellular signaling domain that is derived from the CD3 zeta. And these all components are derived from the T cell receptor. So these CAR molecules inserted into the T cell genome by means of the viral or the non-viral vector, and after it integrates into the T cell genome, then the CAR molecules will express on the T cell surface. After that, the CAR displays on the T cell will find the cancer cells, and then it destroys the cancer cells. Currently, there are 6 single antigen-directed CAR T cell therapy against the hematological malignancies have been approved by the FDA, and this therapy have been showing absolutely great result, and it is showing the absolutely very promising outcome. But not all patient, not all patient. Oh my God, I just run out of the word. I'm so sorry. So not all patients respond in the same way, and some patients, so sorry. And some may have a disease relapsed after getting the single antigen directed CAR T cell therapy. So there are many reasons of disease relapse, and antigen loss mediated disease relapse is one of them. So let's say in this figure here, when we use the single antigen directed CAR T cell therapy against some patient, and this CAR is only effective against the first antigen here, and in some patient after getting the disease relapse, here you cannot see the first antigen expression, and this CAR is no longer effective against this tumor. Therefore, utilization of dual or the multiple antigen targeting CAR T cell could be more effective because it can target the tumor cells even after they have lost one target antigen. So there are ways to tackle the antigen negative relapse by sequentially infusing 2 different CARs in the patient, and each CAR express 2 different antigen markers, and like co-transduction, where we use the different vector that encode for the different CARs. Like the bicistronic CAR, and in one vector they put the 2 different parallel CAR, and it can target the 2 antigen, and tandem or the loop CAR. So one common thing here in this different strategy, we can see the way to express these scFvs. Therefore, the expression of CAR on the dual while targeting the multiple antigen CAR T cell, it is not always easy to express the CAR on the surface. So it needs the complex design, and there should be a sufficient expression of CARs on the cell surface. So there are multiple strategies how we design the dual CAR construct. Normally, we can use the multiple promoter, and each promoter can encode for the each CAR. Likewise, we can use the splitting peptide, and this peptide can differentiate 2 proteins. And we can also use the multiple vector. Each vector encode for the 2 different CAR. However, all of these strategies has some limitation. It causes the large size of genetic cassettes and aids the additional modification of CAR T cells and lead to the low transduction or transfection efficiency. Therefore, we kind of think like why we not to use the bidirectional promoter in the CAR T cell. The bidirectional promoter has a property that can transcribe 2 CARs simultaneously in 2 ways, so that we can reduce the vector size and also increase the transfection efficiency, and also can target the tumor cells even after they have lost one target antigen. So let me give a little bit introduction about what is a bidirectional promoter. The bidirectional promoter is the transcription regulatory region of genes positioned in the head-to-head orientation on the opposite strands. And approximately 10% of human genes are located less than 1 kilobase pair apart in the head-to-head orientation is controlled by the bidirectional promoter. And previously, one of our collaborators from University of Otago, they have tested different eukaryotic bidirectional promoter, especially the elongation factor 1 alpha, which has been already used in the FDA-approved CAR T cell. They tested the bidirectional activity of different eukaryotic promoter, and they found that elongation factor 1 alpha, it has the very strong bidirectional activity in both way. And here you can see that the EF1 alpha can express both RFP and the GFP from the both orientation. So we kind of think like to adapt this bidirectional promoter in driving the dual CAR in the CAR T cell therapy. So what we did is we try to test the different bidirectional promoter like elongation factor 1 alpha, HPGK promoter, RPBSA promoter, and CMB promoter. In this system, we clone our CD19 CAR in the forward orientation, and we clone our CD20 CAR in the reverse orientation. So to analyze the CD20 CAR expression, we just report the expression of GFP here, and then to analyze the CD19 CAR expression, we just use the commercially available peptide. And also noted that our CD20 CAR has a CD28 co-stimulatory domain, whereas our CD19 CAR has 4-1BB co-stimulatory domain. And we made this platform in the Sleeping Beauty vector transfusion. So this, our work has been already published this year in April in Journal for Immunotherapy of Cancer. And I would like to show a little bit what we get from this experiment. Here you can see that out of all the tested bidirectional promoter, EF1 alpha can exhibit the very strong bidirectional activity in both way. Here you can see the GFP expression, which denoted the CAR20 expression. And here this is the expression for the CAR19, and these are the double positive cells. And we also perform the in vitro anti-tumor analysis. Here you can see that this is the bioluminescence picture, and here you can see the total flux graph. So in this here, on day 20 post-tumor analysis injection, you can see here both bidirectional EF1 CD19-20 CAR and the single CD19 CAR treated group can effectively control the tumor burden when compared to the control. However, to our surprise, the EF1 CD19 CAR, which is the single CAR treated group, after a week, it developed the cancer cells again, and then we found that these treated group were not able to control the tumor burden, whereas the bidirectional dual CAR treated group still can control the tumor burden. So there are some advantages of bidirectional promoter, and we highly encourage use this bidirectional promoter in the CAR T cell field as well. So it gives you the coordinated expression, and especially it gives you the synchronized expression of 2 CARs in the both way. And it also gives you the space efficiency. Using a single bidirectional promoter instead of 2 separate promoter can save space in the vector, and it allows for the inclusion of some additional genetic elements in the same construct. And it also helps to optimize the CAR T cell function by including some checkpoint inhibitors or the cytokine or the co-stimulatory molecules. And it is very easy to design, and you can construct many, you can make like very different CAR utilizing only one bidirectional promoter instead of using multiple unidirectional promoters. So what next? Currently, we utilize this bidirectional EF1 promoter by, we just clone this anti-PD-1 SAFE in CD19 CAR. So we are in the testing phase of this anti-PD-1 SAFE, which we derive from the nivolumab, and then we are going to test this system. So when PD-1, we all know from the morning session, so when PD-1 bind to its ligand, this may inhibit the CAR T cell function, but if the same CD19 CAR, if it can express some anti-PD-1 SAFs, then it can block its interactions, and then it may improve the killing. So what's new in this is like people have been doing, and they have been expressing anti-PD-1 in their CAR already. But what makes it different is that normally the people use EF1 promoter like some CARs, and they use some splitting peptide and other like checkpoint inhibitor here. But in some paper, I found that using a different splitting peptide may gives you the uneven expression of your gene, and it may have like some extra modification in your second gene, which may have a problem with your protein expression. So I want to thanks to my advisor, Professor Natia Hirankan. I want to thank to my co-advisors, Dr. Kuramit Supipat, and my co-advisor, Assistant Professor Supin Nakar Tawin. And they are thank you so much for your valuable guidance and the support throughout the journey. And I also want to thank to my international collaborator, Professor Alexander Mekalelen from the Department of Medical Microbiology and Immunology from University of Otago. And thanks to my CUCIT lab members who are already here. So thank you so much, everyone.
Thank you, Dr. Asmita, for interesting talk. We only have time for one question. So any question from the audience?
Thank you for the talk. Elegant solution, need one. My short question is that why did you choose the Sleeping Beauty system for your bidirectional promoter instead of the more conventional retro or lentivirus system?
So as I show you in one slide that the professor from Otago University, Professor Alex, his team already did the bidirectional activity of EF1 promoter in Sleeping Beauty, and from their experiment we found like, okay, this platform is quite, it is working in the Sleeping Beauty, but in their lab, they also try to make CAR20 and CAR19 in the lentiviral backbone, but it did not work. Maybe it needs some more optimization. But we are also currently we are also doing that one also. So hopefully in future, if we can successfully express 2 CAR in lentiviral backbone, it would be like some news.
Thank you.